Okay, so very good evening, and again the extended thanks for organizing this event. Myself, Sudipkumar. Basically, my aim is to work for abiotic stresses, and that cannot be work alone at all, because everybody whosoever associated with agriculture, they have to one point of time has to come one platform, and that platform is called how we can enhance the agriculture yield, how we can make the crops very sustainable with the kind of environmental stress we have and the kind of climate scenario we have. So in this class, on this topic, on this PPT, I will try to focus mainly what are the abiotic stresses, how they are actually impacting the agriculture. At the same time, I will try to touch a few points how we can actually, though we talk a lot about abiotic stresses, how these abiotic stress-based informations can be quantify, or how we can able to know that what actually these abiotic stresses are and how they are impacting our agriculture. So, as everybody know that over the time the climate is changing pretty rapidly, and out of several factors, if we talk a lot about the light is one aspect, then we have wind. At the same time, water play a key role. So these are all factors. They change pretty rapidly over the time. This change over the time, where the plants need to cope up at the same time to grow itself and also look forward for our aim. And as everybody is aware that everybody whosoever work with agriculture, they are always targeting about yield. So with the change in the climate, and these are the few parameters where we can focus. There are large number of parameters. So the plant has to cope up all those external abiotic factors, and these external abiotic factors, they really impacting the plant. So we will try to come across actually how they are impacting the plants and what are the ways get all those informations that effect really leads to the plant change with their output, maybe plant growth and development, and ultimately lead to the yield. So the few stresses, we know that, for instance, if I start from one aspect, starting from the light itself, it's also play a key role, because cloud day might come into the picture that will obviously raise some kind of concern with how the plant can grow, because photoperiod is one more aspect. At the same time, we also look for humidity. Then at the same time, we also look for wind gust, because a gusty wind can really change the whole plant architecture. At the same time, we talk about CO2 level. So that change in the CO2 levels sometimes also affect the plant in more or in more severe way. Then one more important aspect, at the same time, we are taking the quality of water. Though we first instance for the plants, we never ever care about what kind of quality of water we are providing to them, the plant. But ultimately, over the time, we come across with one aspect, that how this actually availability of water at one aspect, at the same time the quality can impact the plant. So these are the few information, the few abiotic stress tags, which I will try to cover in the few upcoming slides. At the same time, whatever we talk, it might be heat, it might be drought, it might be any kind of stress. One thing come in the picture we call drought. And because as everybody know that water is the key component, and because of the presence of water we are here, any change in the depletion level or any depletion in water obviously will impact on not only the plant's health, but also will be impacted. For instance, you might come across the news from last month or so. It's really coming up that one of the city back in South Africa, Cape Town, has no water to use. So these kind of problems will obviously will come, and this for human, but plant has to cope up with this kind of scarcity of water. And what kind of scarcity the plant can overcome? One, it may be meteorological, it may be hydrological, and the third we call as agricultural drought. So there are three terms in a way, but ultimately the aim or the output or effect lies on the plant itself, because plant won't get that water, and once the plant won't have that water, it means the plant will be impacted. So as we are mostly focused on agriculture drought, the amount of water available through rainfall is one aspect. At the same time, we try to use or fill that gap which is there due to the scarcity of rainfall. We can put irrigation in line, and sometimes due to the, again, there are so many abiotic stresses come act together because insufficient to meet the transpiration, and that will become when around the plant high temperature will be there. So water scarcity not only give the kind of stress for the water itself, but also it gives some sort of sense that how the temperature of a plant can increase. Slightly more, I try to put few basic informations here. So actually, why the plant need water? It's obviously everybody know that it's a universal solvent, and it's play a key role in photosynthesis, hydrolysis, and respiration. At the same time, the uptake of any kind of nutrients from the soil or the translocation of any metabolites inside the plant is always being taken care of the plant by the water itself. And then again, there are so many aspects because cells has to make itself safe in such a manner that it can work. In that case also, our water play a key role. And the one most important aspect, as everybody come across their UG or in the early stage, the transpiration is the most what to say necessary evil, but still it's play a key role for the photosynthesis and other activities. So how actually what is being taken, that's being used either for evaporation losses. At the same time, it's also create a transpiration pool, and that transpiration pool just help in uptake of all kind of nutrients which are available in the soil solution in the root zone. At the same time, not only the stomata but other parts of the plant like this lenticular and cuticular and stomatal transpiration is the one where everybody know, but beside this there are some other components which also lose the water and by which the plant can keep itself cool. So everybody focus that whatever we talk about the aim, our aim always remain lies, and even for the same purpose we are here, we will talk about only what to say yield. And yield, if I talk about crop yield, that can be depend upon these three aspects, and out of this one is the key, because if we can understand one aspect, we are exporting large amount of rice to the these Middle East. And at some point of time, it was told by our experts that the amount of rice we are sending to them is not rice, it's actually the water. So we can understand if we can take care of the water, that only not only save the plant, but also several abiotic stresses which are there, which can be overcome. And once they are overcome, obviously the main target or that we call as crop yield is going to enhance for sure. So as it was given by our honorable Prime Minister, sir, that water use efficiency and to be enhanced, and sir has given this tagline, more crop per drop. That has to be done, and that has to be done by just understanding the abiotic stresses, at the same time how those abiotic stresses can be overcome to enhance the agriculture yield as such. So we, as I told, mainly focus for the drought because there are several ways by which the plant can overcome that drought stress. Is try to accumulate as much as water, at the same time is try to make itself slightly more turgid, or as some kind of osmolytes will also come into the picture, and those have a bit of understanding of hormones. Then ABA and auxins play some role, and ABA is the one which actually close the stomata, so the loss of water can be minimized. So out of this whole efforts of the avoidance and the tolerance of drought, there is a few hormones will come into the picture, and whatever is happening at the plant level, ultimately our aim lies always for yield. So we will try to come across that how that yield can be still maintained with any kind of abiotic stress comes. So if we want to understand that one, the key point should be how we can quantify one aspect, and we can find out either it is abiotic stress or not. Until unless we don't have any kind of what to say, if we go to a doctor, until unless he not able to find our symptoms, what kind of problem we have, we cannot go for any kind of medicine, or to be honest, remedy for that one. Same way we need to find out for the abiotic stress. If we able to find out which kind of abiotic stress is there, then we can look several ways. So here I will try to focus how fast that abiotic stress informations can be obtained. So few information, these are the basics ones. I hope you come across with what is genotyping, but among these the most important is the environment, and this environment is actually associated with our abiotic stress. And because, as we know, the genetic makeup of any plant, for instance, is stable, but that cannot be straight away appear in our phenotype, and that being affected by the environment. For instance, extra heat is there, so I cannot expect the long leaves, or leaf may get rolled down, so several effects will come. So we try to focus that how this environment information can be quantified with ease. So one more important aspect, that how we can get those informations, especially for abiotic stress identifications, it can be done by detective. We can detective the plant and can find out the information, but the bad part is that once we cut down the plant, we cannot get the yield from the same plant. Either we need to use some kind of replication for it. For instance, we have 3 replication, R1, R2, R3. We can use one replication to cut down and to get all those information, but yield we need to obtain from R2, R3. That cannot be obtained from R1. And what kind of efforts or the energy, because everything require a cost or resource for this kind of phenotyping or these kind of quantification of abiotic stresses. We need to look for time, labor, and cost intensive. Obviously, large efforts has to be made at this point, and low throughput means it can be done very small pace. So if you want to do for large number, because at this point of time we are planning for going for second green revolution, so the kind of screening or the kind of genetic base we need to have for the upcoming achievements, we need to do in high throughput manner. And one more important aspect, that through this only a few informations can be obtained by this way. Then one more important aspect, everybody talk a lot about that whatever we do that has to be gone to the field. So at the same time we need to look that what kind of genotype by environment, by management, because in the control environment we cannot focus too much on this, but we can maintain the E part. So that kind of changes obviously happen. So our main aim for the field itself to quantify or to phenotype that, to identify the genotypic variation, and that will be lead to we call as a crop breeding pipeline. What kind of extraction we want to put in the crop breeding pipeline, that will become just by the phenotyping informations we are getting from the field. So they, at the same time, what is the field has some kind of advantage and disadvantage for sure. I am explaining that obviously because ultimately our yield will come from field, so it will have variation in terms of soil moisture and the nutrient content. For instance, it was a soil health card was made in the past just to keep an eye that what kind of soil level or what kind of nutrient level we have, those informations are required. At the same time, we need to slightly go into that physiological aspect, that root system architecture, how the roots are spread, how deep they can go. At the same time, we also look for resource use efficiency, how much water was used to give how much biomass. At the same time, we also look that, for instance, we applying urea to the field, how much urea actually, or to be honest, if I go with NPK, how much NPK is being applied to give how much yield. So that kind of efficiency is the key, which we need to understand. But the bad part for this, or the we have some negative point for this, because it will take so many years. At the same time, we need to put a large amount of money for this. So control environment, it can be done very easily. Everything is same, and we can put E, because we can keep the temperature and all the major abiotic stress-related parameters more stable. So that information is slightly more specific what we are getting from the control environment. At the same time, one more important aspect that when we find what kind of changes actually happen all the time with the stress and non-stress plant, if you see here, both plants at this point of time have a different gene expression level, but there are some common genes also. Those kind of informations once come into the picture, then we can look more to improve the crops for any specific kind of traits. Or I can put some other words here that the we up to this stage we are going with empirical breeding itself. Whatever come best out of the we can go for that one. But one more term will come after this, we call as directional breeding. That means we need to plan which kind of trait need to be placed inside the next generation varieties or germ plants, so that can overcome all the abiotic stresses or the climate change scenario which is expected to come in the upcoming years. So overall view like this, my G is here, my the P is here, and these environment play a key role. It might be heat, it might be drought, it might be cold. So all those things come into the picture. And if we take out this from control environment, we can find what is G is equal to P. So these are the information said by Roberto. He said that if we have these phenotypic information, or if I say the quantification of traits, if we can do, that will be the great, ultimately that will lead to the target maybe everybody is planning, that is yield. So but first and foremost thing has to be done, that is that phenotyping has to be done very precisely in stage wise too. And these are the few informations. I introduce one more term for you pupil, that the phenome. A phenome is the study of phenotyping on a genome-wide scale. Whole information of the plant has to be taken at each stage. And now in the last couple of years, the more focused definition come into the picture. We call as phenomics is the acquisition of high-dimensional phenotypic data on an organism-wide scale. I will try to focus how these can be obtained. So few articles published in Nature, like journal, they show that how the plant scientists can shift the breeding into overdrive, means in high throughput manner, how the efforts can be made to just grow more and more faster, good quality crops at the same time, which will give higher yield. So there are several papers comes at the same times. There was a review paper which explained that how these phenotyping can ease the kind of pressure the breeder has, so that they can enhance the yield in upcoming time. These are the few informations. For instance, what how these image based sensors actually work. That may be either reflectance based, it may be absorption, or it may be transmittance. Based on that, we use several cameras, or we call as sensors, and sometimes we call as eyes, because our eyes also have RGB camera in our eyes. So based on that, we try to get information about the plant. And at the same time, when we screen down to say smaller number, then we can look for most expensive like X-ray, laser scanning, or PET scan to get more information. For instance, if we are focusing more on wheat, the root part, then we can look for X-ray and PET scans to find out what is the actually root level changes for any kind of stresses there. So there are ways to find out. It may be electromagnetic based, or it may be chemical signature based. These are the eyes I was talking. We can look for RGB one eye, then we have NIR, then we have hyper spectral whole range from 400 to 2500 nanometer. At the same time, we also look for infrared. I hope during COVID, everybody has seen that when we used to go outside, anybody will come and put a gun on our head. So that used to give our body temperature at one aspect, and it's also giving information how healthy we are. The same thing we apply for the plant itself. And these are the fluorescence. It's also gives some sort of information that how much food formation capability it has. So these are the few informations which are being used to get information about the plant's behavior with the abiotic stresses. And these are the few instruments which are being used for this purpose. And there can be ways, either we can grow the plant inside, and or either we can grow the plant outside, where we have this area is specified, and in this all those eyes or the camera can move and can acquire the information. At the same time, now I hope you come across with the drones, and I hope in this training itself you will come across with the what kind of information drones is also play a key role in spraying. At the same time, it's acquiring information, and most importantly, now drones are being used to identify any kind of infestation in fast way. So that effort has been made, and few information. At the same time, we can look after that how much water use every day. We can put our ports on the load cells, so every minute how much water actually is being used by the plant. Those very specific, very precise informations can be obtained by this one. And one exception, or one more information, you might be thinking that we are doing all those in artificial environment. Several efforts have been made to do in field itself. Here the plants are grown in absolutely field conditions, and all those cameras will go there and will acquire those information, and from image it's evident some kind of change obviously happen. And for instance, any kind of drought or any kind of abiotic stress comes, all those informations can be obtained by these eyes we have, which we have here, and they can scan. And the best part is that whatever accuracy each camera have, that will be applied across all the samples, so there is hardly any chance of error, all those things. And it's one of the facility back in Ikris at Hyderabad. And then we also have some facility back in Adelaide, Australia, and then we have Indra, France. So several facility has been established to quantify the abiotic stress, and that has to be done by image itself. And in India itself, we have one facility in Hyderabad, one facility in back in Bengaluru. Then we have one in Baramati, and the one facility we have in IRI, where we grow the plants. And the best part is that all plants are being put on RFID radio frequency enable identifications. Based on that, all plants are grown. At the same time, they are weighed and water very accurately, and we also get information from the image. And these are the 8 imaging eyes, or 8 sensors, 8 cameras we have, which give informations about the plant temperature, the food formation process. NIR give how much water inside the plant itself, and that all being done non-invasively. Then we have RGB camera. At the same time, we have large range of hyperspectral. These cameras are very new, and they have large scope to acquire large informations, that not only for abiotic stresses. At the same time, we can look for some elemental level. For instance, we have applied NPK in a plant, so how much NPK actually exists inside the plant, all those informations can be obtained by this hyperspectral camera. Then these are the few images how the plants are being used to acquire imaging, and what kind of sensors we have here back in plant genomics center IRI. And at one stage when we acquire those images, we use image processing pipeline to get those informations. And from this, it's evident. For instance, these are my 2 plants. One plant is here, one plant is here. And this plant was stressed, a drought. Drought was given for 8 days. So after 8 days of stress, what kind of changes happen? You could find how many green pixels are here and how many is converting into brown. So all those information, and the traits we are talking about can be obtained by projected shoot area, green area, leaf rolling, caliper length, all those. At the same time, those have some sort of idea of the plant growth rate, RGR, NAR, and all those information can be obtained by just simply taking those images. And it's evident how the image being taken and how the process image comes out after that. One more important aspect, that each stage we also have information that, for instance, I started, it's my control and it's my drought, and I impose drought here. It means I withdraw the water from this point of time, so I could find out what kind of changes actually happen. And this stage-wise information really help us, or really make us to understand what change we need to make at the architecture level or the genomic level inside the plant, that will enhance the abiotic stress tolerance and ultimately will give higher yield. Then these are the image process pipelines from which we can find out which part of the plants were. For instance, if I'm looking for leaf numbers, because they play a key role for the yield itself, and it's still we can find the years itself. At the same time, we can also find out that how plant is growing, what kind of changing, because these are all stages. At each stage from seed to seed, what kind of behavior it will show. For instance, under drought, under N0, so all those informations from this RGB camera can be obtained. And at the same time, we also see that what kind of height is actually changing for over the time. If we don't apply the water, you would see they are not at the same level. It's for it's the control one, it's more, but for the drought is less. So all those information very precisely can be obtained. And at the same time, we can look for AI, artificial intelligence. The kind of data we are obtaining from this all AIs is huge. For instance, one port pass through all those eight eyes, it comes almost 2.4 gigabyte of data can be is obtained, and that contain large informations. So we need, at the same time, we can look for AI aspect in one more way to identify the years. And here from this AI pipeline, you could see artificial intelligence. We use you look once only, and it was old you look 3. Now we are reached in a stage of YOLO version 8. So now we can find out how many years. At the same time, we can also forecast what could be the yield just simply by imaging. Then we also forecast the yield actual and the accuracy for these years. In case of wheat, we able to accurately predict up to 89 to 94% accuracy, how much seed weight will be from the image itself. At the same time, stage wise, as we know, the plant will grow, their leaves will dry, and those kind of information play a key role. Now we talk a lot about stray green trait. So stray green trait quantification required large efforts, but by these RGB camera, now you can find out that what kind of changes, how many leaves are green and how many are dry. So stage wise, we have all those information, and they also help us to look at one aspect, what kind of management we need to take into consideration, and also look at which accession which last up to which last its food formation capacity up to the last, so that up to the last it can keep adding food to the year, so it will enhance the yield. So those informations can be obtained by this image based sensors. And these are the few things to show that how this whole, because I started with one aspect that our work should be not empirical breeding. We should contribute all for directional breeding. And why for the directional breeding? Because we have to face the climate change. Climate change will come. For instance, we have drought, we need to look for root. At the same time, we need to look osmolytes. So there are several factors. It's not only one factor which can play a role. So we need to look several traits in totality to find out actually how that crop improvement can be done. And one more important aspect through this RGB image and the sensors we can find stage wise, because at each stage from seed to seed, at what stage actually how much biomass accumulated, and I can find out. For instance, I know the stage 7 to 8. At this point of time, if we can add more NPK, or if we can provide the kind of irrigation required, so I can enhance that point. So these kind of very precise information, which are the key stages where the intervention can enhance that one. At the same time, we have NIR camera that gives inside information that how much water plant has. At the same time, we can look all those informations in the field itself, and it can give how through the hyperspectral reflectance some parameters and few indices. They are being used to just see how the plants is actually behaving. And from this image, it's very evident that from the image itself we can find out the amount of N, because here we have seen that those plants having less amount of N uptake, those can be seen through leaves. And because if we need to go back to the lab and quantify those, it should require large efforts. But if it's done very fast through the image, I think it can lead to the quality informations which can be put in the breeding pipeline in faster way. So these are the few. It shows that these accession, what we are showing, Chotil Herma is the one which is highly nitrogen deficient. So those kind of informations can be obtained by this hyperspectral camera. At the same time, the food formation capability can also be seen. And as we know, any kind of abiotic stress, for instance, come, it will affect the plant for sure. That effect can be quantified with this fluorescence camera. It gives the photosystem to activities high or low. And once we establish this kind of correlation, because though those informations come late, but through the information we are getting from RGB or these fluorescence camera, very precise, and that gives the strength to give those material to the breeder, which can be really overcome those kind of stresses and can enhance the yield any fast way from this. At the same time, as we know that the whole leaf here, in this one we have two plants. One plant is the wild type here, one plant is transgenic. So transgenic plant can overcome so much of drought, because here leaves are still active. Here in wild type, the plant has dried. So all those informations can be obtained by this way. At the same time, that infrared camera, as I told, also give the informations. At the same time, MRI and PET can be done. For instance, if want to go deep into the what kind of changes actually happening at the root level, and for instance any kind of stress comes, so what changes actually happen inside the root, we cannot see from outside. But by this camera it's very easy. For instance, what is the root angle, how deep they are going, and how much they grow. So all those informations from the root itself can be scanned through MRI and PET. And this is the ultimate equation where for which everybody is working, that we need to enhance the yield. And in that one important aspect lies water use efficiency. And some effort has been done back in ECRISAT itself. They take the weight every day, but it's a very tedious job to take the weight and to find out the biomass. In our plant phenolics facility back in Nanaji Desmukh, we have a weighing and watering station. Every day plant will come, weighing will be done, and the amount of water it has used in the last 24 hours through transpiration will be added. So in the last, we will be able to find out how much water was used for giving how much yield. These are the few good publications which comes out, which show that how this high throughput phenotyping can detect a drought. And once we have that drought, that's information is being used to improve the drought tolerance in barley. Some effort has been done to analysis, especially in barley, because effort was done back in 2014 and 15 in China and Germany. At the same time, QTL mapping, because if you are aware of the QTLs, we are only going with end QTLs, but through this stage wise information will be available, so it we can find out all those information at each stage by this image based information. And at the same time, because sometimes this kind of scenario may happen, because in the middle of the life cycle the plant will behave in a differently, and in last it will reach like this. But what kind of strength or what kind of gene level expression here which give those strength, that information only can be obtained by the stage wise information if you use image based features. And then dynamic QTLs, those are from the plant breeding side. They might have some short idea that the QTLs we are talking about for the end only, but at the same time always gene will express and down express. This is always there. But if we able to pick those which actually over express or express at the time when the actually stress there, so that will be really helpful for the upcoming generation to breed good quality excisions which can overcome those stresses. So again, to take home message at this point of time, drought always spec will come into the picture. Soil, water, all those things has to be estimated. At the same time, if I did not talk too much about the temperature stress, but still they are more or less correlated, because when high temperature will be there, the amount of obviously water is going to be less. That will be affect and in the same way. And the third kind of stress, though it's exists in the eastern part or the northeastern part of India and some part of Australia, but still salinity play a key role, especially for wheat and barley. So that aspect has to be taken into consideration. And the understanding about these three stresses, again I will say, the kind of effort we need to make, that should not be a small one, that should be big one, and that should be done in high throughput manner. So the overall summary for my talk here, that because everybody talk now digital agriculture, because until unless digital agriculture won't come into the picture, it will be difficult to use the old tools to get all those information and improve the things. But still, with the high throughput phenotyping, large data analysis capabilities, growth analysis parameters, now we can reach up to a stage where we can plan that what kind of target accessions, target variety we need, we can plant those and we can breed for that one. So I like to especially thanks all my mentors and team who just work in behind and support me to give all those informations to you. Yeah, thank you. I can take a few questions for now.